We are very excited to have Dr. Hassebroek here today. She is the head of genome engineering at Stowers Institute for Medical Research in Kansas City. Her research background spans molecular biology, genome engineering, epigenetics, and chromosome biology, with extensive experience using CRISPR-Cas9, imaging, protein analysis, and functional genomics approaches to study mitosis and centromere regulation. Hassebroek earned her PhD in molecular biosciences from the University of Kansas, where she developed expertise in cell culture microscopy, genome editing, and biochemical assays. Prior to her current role, she completed postdoctoral training at the Stowers Institute investigating molecular regulators of the epigenome. We are very, very excited to have you here with us today. And whenever you're ready, please feel free to get started.
Awesome. Thank you so much for that kind introduction. I am really excited to talk to you guys all about how AAV donor delivery has really transformed genome editing in our institute and across many, many different model organisms. So Smitha just gave a really nice introduction to what I do. If I can advance my slides. Um, but I wanted to start off by telling you a little bit about my path to where I am today. So I started at the University of Missouri in Columbia, where I received a bachelor's in biological sciences. And this is where I really got my passion for research, where I spent 3 years in an undergraduate research lab studying different neuroscience questions. And it was from that experience that I really know that I wanted to go to graduate school. And so after our graduation, I joined the University of Kansas graduate program. And some people will call this like a traitorous act because these are two schools that are kind of have a history of being at war with each other. But it was a wonderful transition, and I really enjoyed my time at KU. I spent a year rotating, and then I joined Yoshiaki Azuma's lab in 2016. And I focused on mitotic regulation and how post-translational modifications can mitigate different chromosome binding proteins. And so it was in that lab that I really became obsessed with CRISPR. This was the time where that paper came out, where they started showing how to use it in mammalian systems, and this really kind of lit my fire. And so I asked my PI, can I try this in our lab? And I spent a lot of time and energy trying to pioneer CRISPR. And I basically figured out all the ways that you should not do it. And then finally, I did have some great successes, thankfully, and that kind of spearheaded my post or started my postdoc after that. So I transitioned into Jen Gerten's lab, where I studied transcriptional regulation in micronuclei. But I never really lost my passion for CRISPR, and I knew that I was very interested in tool development and kind of assisting research. And so I really wanted to kind of stay in the technology center side of things. So thankfully, I was hired as a scientific project manager after a 3-year postdoc. And this helped me to kind of get integrated into our core system. So we call them technology centers. And this is where I would really liaison with students to help them connect with the right people in our core facilities. And I also got to help sort of plan and execute different CRISPR experiments with those students, which I never kind of lost sight of CRISPR. And then in 2024, I was hired as the head of genome engineering. And before I go too much into the actual stuff that we do, I want to talk about the Institute itself because Stowers is an incredibly unique place. We're really lucky that this is an environment that really encourages discovery-driven science. And we were given a very generous endowment from Jim and Virginia Stowers. And so we are privately funded and don't have to contest with all of the different financial struggles that are going on in the country right now. The Institute's unique funding structure really helps us to be able to focus on innovation and do a lot of different cool things. And so I hope that you kind of see the different types of freedom that I've been given to do science in today's talk. We have 20 different PIs. We have a graduate school and a thriving community of postdocs. And no two PIs are the same. So we have a really rich and diversity of people and also in science. I have the absolute blessing to have all of these people on my team. They have been incredible in kind of spearheading a lot of these projects. There's a wealth of expertise and knowledge in this group, and I'm just really lucky to be able to work with them. So all of the things that I show you today would not be possible without this group of people. And so, um, one of our major jobs is to take a biological question and use CRISPR to try to build the genetic tools to answer it. And I know that this crowd is probably not one that needs a reminder about what CRISPR is or how CRISPR works, but I will use a lot of acronyms, and I just want to have everyone on the same level playing field. So I'm just going to do a quick introduction to CRISPR-Cas9 editing really quickly. So of course we have a Cas9 molecule bound to an sgRNA, and this molecule together is called an RNP. This can be introduced into cells where it will cause double-stranded breaks, and those can be corrected by two different pathways. So we have our non-homologous end joining, or NHEJ, which essentially pushes those two deletions together and introduces a deletion or an insertion known as an indel. These are going to cause a frame shift where we'll have an early stop introduced, and this will cause no gene output. So we will cause a gene knockout. An alternative pathway is called homology-directed repair. And this is a really important pathway to people doing CRISPR because this is how we can introduce our edits of interest. So through this double-stranded break, you introduce a molecule, a donor molecule, which has homology to the place you created the break and your insert of choice. And this will become repaired in a small subset of cells. I want to emphasize that most cells are going to go through this pathway, and you really want them to be going through HDR to be getting your insert of choice. So at the Institute, we have a really large plethora of different types of organisms that we edit. So we and different types of edits that we do. So some of our most common ones are causing gene disruptions. So this is by causing a knockout. These are relatively easy to do across all of our model organisms. We also have worked in a lot of different transcriptional regulation using CRISPR-I mostly, but also CRISPR-off. And largely we'll do knock-ins where we are introducing a fluorescent tag, an affinity tag, floxed alleles, or we might be recapitulating a human disease by introducing a SNP. And we'll also introduce conditional alleles, and we usually do this at a safe harbor locus. And these are three that we use regularly in the different models that I'll talk about. So I've been mentioning how often we get to use a diversity of organisms, and so these are just a few of the things that we edit. So this is typically we'll be doing edits in cell lines, human cell lines. Mouse, of course, and zebrafish. So those are sort of our three workhorses. But we also do create CRISPR tools for people to do Drosophila edits, as well as a golden apple snail and Nematostella. This is an anemone species that is shown here. And we're hopeful, we're designing some stuff right now to try to edit some coral species that we have on site that are due to spawn this fall or winter. But there's an ever-growing list of PIs, fellows, and postdocs that arrive at the Institute. So the list of organisms is constantly growing as well. We just recently introduced David Stern to the Institute, from Janelia, where he will be. We are hoping to edit some aphid species that he has. We have a postdoc who recently brought Ceratopsis, which is a immortal jellyfish. They have a very cool life cycle. And we have a fellow who works on a lot of different flatworm species, like this one shown here. So we have to stay very flexible because our list of organisms is constantly growing. And that's why efficiency really matters to us. We do around 70 edits per year. And of course, we're always trying to drive researchers' questions forward and help their science to move in a forward direction. And so we don't want our timelines for generating organisms to hold them back from answering their questions. And so one thing that we've really found that is to be true is that there's no universal donor strategy. Every system really requires specific optimization. Just because something worked in cells does not mean it's going to work in an embryo or a different organism. And really where we have found the toughest struggle is in the actual delivery of the donor molecules. And so that's what I'm going to really focus on today is this delivery. And so just for the first introduction to one of our organisms is human cells. We use a lot of different human cells on site, and our typical RNP integration method is electroporation, and our typical donor molecule is a single-stranded DNA donor. And depending on the type of cell line that we use, uh, it can be very, very easy to get edits, or it can be very, very difficult. And I'll go into some of those more difficult stories in a few slides. But the cells do tend to have consistent HDR integration, so we are happy about that. So we have variable success rates depending on the cell lines. Next is zebrafish. So of course we use a microinjection method to introduce our DNA to the cells, or our RNPs to the cells, the embryos. And we use a donor molecule, which is a biotinylated PCR product. This is a double-stranded DNA molecule with biotinylated ends, and these really help to protect the ends from exonuclease activity. We can generate knockouts relatively easily by introducing an RNP with a guide to the locus of interest. But generating knock-ins is extremely difficult, and it's quite basically numbers intensive. So zebrafish have a really unique biology in early embryogenesis where they will perform about 30 cell cycles in about 30 minutes. So they're really trucking through those cell cycles extremely quickly, and I don't think that they rely very heavily on homology-directed repair to correct double-stranded breaks. And so it can be very difficult to get large knock-ins in these fish. The next is apple snails. So we use a microinjection method for this as well. We were the first actually to ever do CRISPR in apple snails, and it was really exciting to generate our first knockouts. The time it took an incredible amount of time to optimize this, and one of my team members worked really extensively with a postdoc here who has recently gone off to be a PI in California. But the effort level was really high. But we can generate knockouts. We are currently working on optimizing donor delivery strategies to find out exactly what will encourage these embryos to do HDR, because right now what we're seeing is kind of a very jumbled integration, and so we're not sure if they're using a standard or a human-centric type of homology-directed repair. And so this is kind of a key point to mention, that non-traditional organisms really require a specific tailoring strategy to get edits to happen. Next is Nematostella. So this is that anemone species where we also utilize microinjection. And so to see the embryos, we illuminate with bright field, and then we cover the bright field to actually see that we're integrating DNA into them by this fluorescence molecule, which is co-injected. These also utilize a PCR product as their donor strategy. So we use double-stranded DNA for their donors as well. We can be pretty successful generating knockouts, but it is on the more difficult side to generate knock-ins. It has been successful, but it takes a lot of injections and a lot of numbers to, it's quite labor intensive to get these knock-ins. And we have noticed that F1 transmission is a little bit low for these guys as well. So next, I'm going to talk about how we used to do mouse embryo CRISPR, which is either by microinjection, which you can see on the bottom, or electroporation on the top, which is not the most exciting video. Of course, you all are probably familiar with this, but we were integrating either a plasmid or a single-stranded DNA along with our RNPs. And this was moderately difficult. We could do this, of course, but it took a lot of embryos and it was, again, quite labor intensive. And so that really brought us to question. Oh, my slide is gone. It brought us to question. So how can we get this? Oh, excuse me. I'm okay. Here it is. So we this is a graph that's really exemplifying how difficult this was to do and how our integration rates were not the greatest. So you can kind of ignore all of this on the side. This is just different genes that we've been editing, and their edits are alongside them. The MI indicates microinjection, and the EP indicates electroporation. Where we saw that with microinjection, we had about a 16% integration rate. And with electroporation, we had about a 40% integration rate, which was wonderful. But the biggest limitation is this point that I'm making here, where we have a huge size limitation for electroporation, where we can only integrate things up to 500 base pairs. And because a lot of the times when we're doing integrations, we're trying to introduce a fluorophore, which are at minimum about 800 base pairs with no homology arms, electroporation just did not work for those molecules, for those kinds of insertions. So on average, we would need to repeat an experiment about 4 times for microinjection and about a little over 1 time for electroporation. And on average, we would use about 762 embryos for microinjection and about 188 for electroporation, which is electroporation clearly is the winner of this slide, but I want to really drive the point forward that this was only true with donors that are 500 base pairs or smaller, which for some affinity tags, that's great. But for fluorophores, that's just absolutely not big enough. And so we started to ask a different question. How do we improve donor delivery for things that are larger than a small insertion? And this led us to adeno-associated viral donor delivery, which is a adeno-associated virus is a non-pathogenic viral vector. So this is a BSL-1 level virus. It's a really, really efficient donor delivery system, and it allows you to package single-stranded DNA templates, and when they're delivered to the nucleus, they become double-stranded within the cells, which is an incredible molecule for doing homology-direct repair. It improves integration because you have a higher efficiency of donor delivery, and you're also being more gentle on the embryos. So microinjection comes with even the best injector will have death because this comes with a lot of biophysical force. And so doing a co-culture has been a lot more gentle, and we have a lot higher 2-cell rates, which has been great. It is also a very consistent delivery system, and so we know that we're going to kind of get the same delivery every time. And the really important part is that you get a high donor copy number within the cells and within the nucleus. And so this is where all the things are happening, of course, you're getting your double-stranded breaks in the nucleus and you're doing homology-direct repair there. And so you really want that concentration to be high where the repair needs to be happening. So this is just a little schematic, which just emphasizes that AAV delivery has puts your molecule, your donor molecule, where it needs to be at the time that it needs to be there. And so our mouse embryo introduction looks a little bit different now, where we collaborated with our EM core here, and we got a picture actually of our viral molecules for some fun. So we all of the small circles that you see in this SEM image are actual viral particles, and this was a pilot prep, so a lot of the chunks are probably FBS or cell debris. And so what we will do is we culture our embryos in about 10 to the 8th virus in the morning. And in the afternoon, we will go and we will electroporate these embryos with our RNP. So the donor is actually already in the embryos when the Cas9 and guide are delivered. And our efficiency has been incredibly high using this strategy. And I'm going to talk about that a little bit on the next slide, where you can really see our integration has gone up to 31%. And this is for anything up to 4.7 kb is the packaging limit for AAV. But excitingly, we can also use a split AAV system, which has had exactly the same success rate for a much larger donor strategy. So this is a 6.8 kb insertion that we split amongst two different AAV packages, and we co-infected the cells with those two packages, and the embryos did the rest, and we got this on our first try, which was really exciting. The average number of times that the experiment needs to be repeated is actually technically 0. This is a misnomer, but we actually only have to do the experiment one time to get 2 founders, which is really exciting. That really drives our number of embryos used way down to 167. And I think this next slide really emphasizes how much AAV has changed our efficiency for the mouse work. So just to emphasize, our integration efficiency is 31% as compared to 16%. And as a reminder, the electroporation efficiency is higher, but only for molecules that are 500 base pairs or less. And so since we're usually integrating larger items, we really focus on this comparison between our 31% with AAV versus our 16% with microinjection. Our number of embryos used, again, is hugely reduced, which is incredible compared to microinjection. And our number of times where we need to do the experiment is reduced a lot here, from 1 time or from 4 times to about 1 time. And so this has really allowed us to make a lot more mouse strains a lot more quickly, which has helped the Institute as a whole to ask new and defining biological questions. And this is it's just been a really fun experience the past couple of years doing this. And so really CRISPR has, the field itself has shifted quite a bit from the bottleneck used to be cutting DNA. So your guide selection was very tedious, making sure that you have a stable RNP and having efficient cutting, all those things in the past were more tricky. Those have all been really standardized, and the algorithms have gotten incredibly good for guide selection. And so those are no longer the bottlenecks when doing a CRISPR experiment. The trick is HDR integration, large donor delivery, and utilizing difficult cell types. And so this is not necessarily a problem that we have solved in cells. We have inhibitors that really boost HDR, but even boosting HDR in a cell that doesn't have the donor is not really going to help you too much. And so in these really hard to edit cell lines, we kept kind of hitting our head against the wall, and we decided, well, let me tell you a quick story first. We work with a cell line called retinal pigment epithelial cells. This is a really important cell line to Jen Gerton on site. She was a part of the T2T consortium, which means that she was on the team that helped to do the genome assembly from telomere to telomere. So to fill in all of those alpha satellite regions, repetitive regions, especially for her, that was important to have the centromeric region assembled. Now, that was no small feat, and they did that first in CHM13 cells, and then they moved into RPE1 cells, where they now have an assembled T2T genome. And this is important because this is sort of their workhorse in the lab. So because of this full genome assembly, the Gerton lab has really emphasized use in the RP1 cells for asking their questions. So we were stuck with them, and we really wanted to find out how can we fix this problem. So the problem is that we can very efficiently cut the DNA, and even though we have nearly 100% at cutting, we had nearly 0% of our integration, our HDR integration. So we used RNP delivery and a single-stranded DNA donor, and we used electroporation to integrate that. And we had nearly 0% of the cells were M scarlet positive in this FACS plot. And so we tried all of the different inhibitors. We tried a lot of different, we tried many different electroporation uh, types. We tried different, tweaked the settings. We tried different electroporation uh, actual models, and we tried many different chemical transfection methods. And none of these really improved our donor delivery efficiency within the RP1 cells. And so I was thinking we're having all of this success with our mouse embryos. Why don't we try AAV delivery for the cells? And so we found this is the same gene. Um, the first experiment that we did, we had a plethora of clones that were positive, and we actually stopped counting them at some point because we had genotyped 37 positive clones, and a good portion of them were homozygous, and a good portion were heterozygous. And I said, okay, I don't think we need to keep genotyping these. We have plenty to freeze back and to use. Um, and so we went from absolutely zero to too many, which was a great problem to have. We also had a different gene that we had spent probably a year trying to integrate into the cells. This is a locus that we were trying to edit as a parental line for introducing an AID. So we were trying to just introduce the ubiquitin ligase gene or into this RCC1 locus. And this experiment, although it had less clones, this was actually our first AAV experiment, and we learned that um, after co-culturing, the cells are more sensitive to FACS sorting. And so we actually, um, even though we had a lot of cell death after sorting or less recovery of clones, we still had 4 clones, which was plenty to move forward with our experiment, which was exciting. So from this experiment, we actually learned that if we give them a week of recovery with just a couple of passages, we get full recovery for FACS. We also did this in a couple of other gene sites as well as a different cell type altogether. So this is K562 cells, and this was kind of a large insertion. And so I thought, hey, let's try AAV, even though it was kind of prescribed to not work for K562 cells. We had great delivery and we had great clonal recovery. For all of these, we used the AAV 6.2 and we used the pilot packaging scale. So because we were having such good success with cells, multiple different cell types, as well as mouse, I decided I would like to systematically test AAV on our other model organisms as well and our non-model organisms. And so we purchased this AAV testing panel basically from Vector Builder, and this gives you 13 different types of different serotypes with a CMV EGFP packaged inside. And so our first test case was with the Nematostella. So this is the system where we have to do microinjection. And even with microinjection, you have to do many injections across many embryos. And still you have pretty low numbers of founders and low levels of F1 transmission. And so I thought, let's go ahead and give this a shot. And I'll apologize automatically for the data quality, just because these came off of the press basically this week. So this is a really exciting finding. So our control Stella embryos look like this. We kind of have like a low bright field setting on, so they're very dim. And when we co-cultured with AAV2, 3, and 4, we do see very strong GFP fluorescence at 24 hours. And this was really exciting. Again, this is a very hot off the press type of data. So we're going to do some follow-up experiments and sort of titrate our amounts of virus used as well as our timings for this viral introduction. And if we can recapitulate this data, we'll be excited to pick a serotype, package an actual donor, and try to do a CRISPR experiment in these guys using AAV to test if this is going to be a strategy to help our HDR improve further. And so, of course, the questions that we ask every time are, can AAV enter the embryos efficiently? So this is suggesting that it can. And does co-culture improve donor availability? So this will be the question we ask when we pick a serotype and redo the experiment with an actual CRISPR experiment. And do we see GFP expression broadly? How long do we see it, et cetera? This is all important questions to ask. And eventually, could AAV support HDR in cnidarians? And this is all really exciting because if we can improve AAV delivery, if that indeed helps us, it really will broadly expand our scalability for HDR in genome engineering in general across our different models. And so, of course, as we improve delivery, we expand what's biologically possible. And a better donor delivery really enables faster iteration between designs. So instead of waiting months to have your animal do the experiment and realize, oh, I want to change my fluorophore, I want to change my tag, because we can make animals more quickly, we can iterate faster. It reduces our founder generation timelines as well as the amount of animals that we have to use to generate founders. And now we have access to previously difficult biological systems. So, like I showed you, we have a plethora of different organisms and models and non-model organisms or newly emerging model organisms that are going to be really important to test. And if we can do CRISPR in them, that really opens up a ton of biological questions that people can start to ask. And so, with that, I wanted to thank everyone for their attention. I need to thank my team. All of the things that I showed you would not be possible without this incredible group. All of my collaborators within the Institute. So this is a very small subset. This is mostly just the data that I showed, but we collaborate with almost every lab at the Institute. And we're really blessed to have a ton of technical support. So we have, I think, 15 technology centers on site. And that is a lot of help with actually maintaining the animals, keeping them alive, getting us embryos, etc. So we would like to thank specifically rodents, fish, and inverts, as well as the Cells, Tissue, and Organoid Center for helping us do these integrations into the cell lines. And of course, I'd like to thank the cytometry team for letting us do FACS and helping us do single cell sorting. And the automation team helps us a lot of the times to do our genotyping. And lastly, I'd like to thank Vector Builder for the incredible communication and help that they have provided to provide all of our viral donors, and of course for inviting me to speak today. So I've put my email up if you guys have any specific questions, and I'd love to answer any right now as well. So thank you. And with that, I'll take any questions.
Thank you so much. That was incredible. And especially as a person that predominantly worked with non-popular model systems, this was very exciting too,
Oh,
actually.
good, good.
We do have a couple of questions. We can start with, are there any concerns associated with having the AAV donor present in the system throughout the development?
So far, we have not seen that to be an issue. It is possible, but a lot of these animals are going to be outcrossed, and so I would think that any kind of issues that you would see in the founder would hopefully be bred out and diluted out.
Cool, thank you. For the split AAV used as the knock-in template, how were the AAV genomes designed? More specifically, were the AAV rejoined through homologous arms or an AAV link-like design?
That is a great question. I should have gone back and looked at that. That was over about a year and a half ago now. And I can tell you actually, the people who helped us design, because we had never done that design before, was Vector Builder. So they have an incredible support staff to help you do your design. And so we basically chatted with their support team to say, okay, we have this really large integration. What split strategy works the best? And I believe that we had I'm gonna see if my team will teams me about this, and then I'll answer that question. I'll come back to it if I have to. But otherwise, definitely reach out to Vector Builder to get that support because it is there.
Thank you so much, and thank you for the shout out. What's the reason for high HDR rate using AAV, but nothing using single-stranded ODN in the cell line?
So we have incredible success in like K562s, for example, or 293s because they electroporate or transfect really well. And so getting a single-stranded molecule into those cells is really not difficult. And so I think that that's why a lot of people have, and we do have success using single-stranded ODNs. The issue becomes with cell lines that are not transfecting efficiently, the larger your molecule is, and a lot of times we are trying to introduce a fluorophore or a lot of things plus an AID tag. Uh, your integration rates go down. And so I think that the homology-directed repair is just not happening because the donor is not there in the nucleus at a high enough concentration. So, a lot of times our integration rates, especially in RPE1s, is like 0.1% sometimes.
Wow. Thank you.
Yeah,
That's really interesting.
terrible.
Are there any possibilities on random integration of AAV in the genome? Any analysis done on that?
We do always keep in mind that that is a possibility. It's less likely because you're delivering a single-stranded molecule, but um, we do keep that in mind, and we always do a copy number analysis to double check that we are homozygous, just have two copies. Um, yeah, that's a great point. It's always good to
Thank
check.
you. We have a technical question. Which medium was used for the AAV co-culture with the nematostella embryos?
Oh, with the Stella embryos, they were actually in their salt water, I'm pretty, or in their typical water, that we just put them into a droplet of their water, and then the AAV was added to that. So they were co-cultured overnight, I believe, and that's when we saw the expression. But again, if my team members are watching and they can tune in, I see someone typing now, so they'll correct me if I'm wrong.
Thank you. Are you using a consistent HA length regardless of insert size?
I have the split AAV answer really quick. So we had 1 kb overlaps, if that helps that person, hopefully. Yep. And then what was this question?
Are you using a consistent HA length regardless of insert size?
That's a great question, and I will say that varies with the type of organism. So, our typical homology arms are usually a lot smaller than they used to be. So I think our typical is like 50 to 200 base pairs. It's smaller for zebrafish. I think for human cells, it's usually about 100. But of course, if it's just a SNP, we can get a little bit smaller. If we're trying to do smaller integrations, our strategy is going to be to try to order an ultramer, which is to keep the donor as small as possible, just because it gets here the quickest. But for like a fluorescent molecule that's 800 base pairs, I'm gonna go to the 200 base pair homology arms. So yeah, it can scale with size, but it more scales with organism.
Thank you. We have quite a few questions, but we also have quite a bit of time.
Oh,
So
good.
this is our next one. What do you think about concatenameric integration using AAV? Do you verify it and how?
Okay, so this is a great question that I actually forgot to bring up during my talk, and I'm glad that you asked it. So we are aware of the risk of concatemerization, and the way that we have tried to mitigate that is by introducing a guide RNA in our embryos, which is complex with Cas9 that targets the ITR region. So we actually target and cleave the ITRs, and this has helped us to mitigate concatemerization. And we check that by long read sequencing to make sure that we are avoiding having multiple repeats or concatemerized molecules.
Okay, thank you. Do you have any plans to develop any novel AAV serotypes to be used
Oh,
more effectively to
this would
target?
be fun. Yeah, this would be very fun. I think as we start to delve into the non-model or the new model organisms, likely we will have to get creative with the serotypes. We do not have a facility on site that does AAV packaging, so we would probably have to collaborate with Vector Builder to do this. I was pretty excited. I was actually very surprised that we had such nice GFP signal from our three serotypes that we tried. And this was only we so far have tried the first half of the library on the Nematostella, and we're trying the second half. I think we'll have that data today or tomorrow. And so it will be interesting to see if there's more overlap across evolutionary time than we expected to see. But of course, if we kind of get to the point where we're not seeing any GFP signal, we will try to make some different types of serotypes. Yeah.
Okay, cool. Thank you.
Mhm.
Are you only working with cells, human cells, that grow from single cells for cloning?
Typically, yes, we are almost always going to take we do bulk analyses every so often. Some researchers just need quick data and they don't mind having sort of uh, a bulk population of cells. But typically, we are going to do a CRISPR integration and then we're going to take it to single cells and grow from there. So we have clonal populations, and we always tell people it is best practice to have at least 3 clones, if not 5, that you can compare and make sure that they're giving you the same biological output. Because, of course, when you do CRISPR, there are background mutations that can happen. So, yeah.
Hmm. Thank you. We have another serotype question. Have you compared the efficiency of AAV serotypes in mouse embryos? If so, which serotype is most efficient?
So we have not tried a comparison because AAV1 works so incredibly well. So I would not even go through the effort of trying to compare. Um, AAV1 is the tried and true for our embryos. I know people use, I think, 6 and 9 for doing different in vivo, like in the mouse type AAV delivery, but for our embryo co-cultures, we are using 1.
Okay, thank you.
Mhm.
1 kb overlap is for both up and downstream homology arms. That's the question. And the size of the homology arm related to GC percents, position of locus, etc. Which one is more important?
So I think the 1 kb overlap that I was discussing was between the two different donor molecules, because remember, this was so large that we had to actually split it into two different donors. And we wanted them to recombine in the nucleus and then use that to do homology. Then the cells would use that to do homology direct repair. Um, I believe our homology arms for that mouse were actually 500 base pairs, flanking on each side. So those homology arms for the actual genome, and this overlap was for the actual donor molecule, if that helps. And then
Mm.
what was the question about the GC content?
Size of the homology arm related to GC percents, position of the loci, etc.
So, that's getting a little bit more into the weeds. Typically, we don't focus too much on GC, and typically you're kind of limited to where your guide is. And so, and if you have a particularly GC rich region, it might be a little bit more difficult to edit. So, for example, that rDNA plot that I showed, we used the AAV and K562 cells. That was extremely GC rich. But I don't think our arms were longer than 200 base pairs. And we didn't have any trouble actually getting that homology direct repair to happen. So um, I think again, it's really more the limitation is not necessarily the CRISPR design. It's actually having your donor molecule in the nucleus at a high enough concentration to be used as a donor. That's the trick.
Awesome, thank you.
Mhm.
Can you give the sequence of the sgRNA that cut ITR?
Oh, I would be happy to share that. We have not published that. If it's possible, please send me an email. Take my email, and reach out to me because I would love, yeah, personally, I'll be happy to share that, but I do want to make sure that there's no weird like MTA things for that since it's not published.
Okay, that brings us to the end of our questions. Unless somebody has any last minute questions, we maybe have a couple more minutes, so please feel free to type them in. And thank you so much for all of these questions. They were incredible.
Yeah.
Let's see. Yes, I don't see any more
Okay, cool.
questions. So thank you so much again, Victoria, for this
Yeah.
wonderful talk, and especially appreciate you showing data that is less than a week old.
Yeah, we're always on the cutting edge here, constantly moving forward.
Yeah,
So,
that was absolutely incredible. Thank you so much. And
uh-huh.
thanks everybody for attending. And we hope to see you at our next Gene Delivery Seminar Series talk.
Thank you.